Отже, сьогодні я дуже радий вітати у нас Олександра Ісаєва, професора Carnegie Mellon University. Олександр закінчив у 2002 році Дніпровський національний університет, потім захистив PhD у США і зараз працює в галузі теоретичної хімії, медичної хімії та використання якраз штучного інтелекту в різних галузях хімії. От їх сьогодні представить нам свою лекцію. Прошу, будь ласка.
Дякую, пане Олександр. Та також мої вибачення за проблеми з time zone. Привід годинників на літній час – це зло. Перш за все, я хочу acknowledgement додати до усіх студентів та працівників моєї лабораторії та усім агенціям, які підтримують нашу роботу та плідну співпрацю з Гінамін Space та Юрієм Морозом. На жаль, я поїхав з України майже 20 років. Та англійська – це мова моєї лабораторії та це мова науки. Тому ми спробуємо провести цю лекцію на англійській мові. Але, будь ласка, якщо щось, які є запитання, то, будь ласка, запитуйте, та я буду радий цю лекцію зробити діалогом. Якщо ви не були в Америці, Carnegie Mellon знаходиться в штаті Пенсильванія. Це East Coast, біля Вашингтона, Нью-Йорку. Це закреслений штат Пенсильванія. Це як він виглядає як кирпич. Та Піттсбург десь на західній стороні Пенсильванії. Дуже інтересно, але Піттсбург має такий вайб, як Дніпро. Стара індустрія, сталелитейні заводи, кілька університетів та велика єврейська община. Університет Carnegie Mellon – це частний університет, один із топових університетів у Сполучених Штатах. Це наш кампус зліва. Це здання – це наш Department of Chemistry and Physics. Та цікавий факт, якщо ви бачили трилогію Dark Knight, це «Темний лицар» по-українськи, то знімали на кампусі та у Піттсбурзі. Ви можете, це мерія Готэм Сіті. Now, when we ask a lay person a question, so can you imagine a chemist? And people usually think about chemists, some suspicious, people who mix liquids and do some strange experiments, colorful liquids, fumes, and things like that. However, in practice, chemistry is really diverse. And in fact, the chemistry that I'm working as computational and theoretical chemistry, our lab is actually a normal office. It looks like that. And many of that is because we use computer simulations. And the progress in our field actually due to this chip called GPU, graphic processing unit. So if you game on a computer or a PlayStation, so these are chips that renders your graphics. But also those chips are behind the current revolutionary AI. And in Pittsburgh, we have Pittsburgh Supercomputing Center, and it's one of the largest supercomputers that has, those chips to do not gaming, but various types of computer simulations. And also, as we go further, chemistry is also being robotized. So the advancement in robotics and automation is probably going to be the next step. Now, when I was defending my master of thesis back in Dnipro, my thesis committee were skeptical about the perspective of theoretical computational chemistry. So many years later, so there's a print screen from the news of the Royal Society of Chemistry in UK. And what the incoming Royal Society president says that in 15 years' time, no chemist will be doing experiment at the bench without trying modeling first. So this growing appreciation of the modeling and informatics is really growing. And you probably have seen all in the popular media, but the rise of the AI and smart technologies are all around here. So there's a couple of examples, not from popular literature, but from the scientific news. So here on the right is the chemical engineering news. This is a news magazine of the American Chemical Society, rise of the smartest machines. Here on the left, again, Royal Society of Chemistry, wanted synthetic chemist, human need not apply. So I'd like to motivate my lecture by this quote. And this is really a thought-provoking quote. It is attributed to Derek Law. Derek Law is one of the most famous blogs to the science translational medicine. He's a pharmaceutical industry veteran. What he said is that it is not that machines are going to replace chemists. It is that chemists who use machines will replace those that do not. So it's really thought-provoking quote, and that's definitely motivates our work. Now, if I go, and you probably all of you seen this new shiny system called ChatGPT. So this is essentially, it's a chatbot and powered by this AI engine. And you can ask the machine, what is own opinion about the AI in chemistry. And this is surprisingly accurate quote. And I will not read it fully, but what is also important, AI can help chemists explore vast complex chemical space, find novel and diverse molecule, desired properties and activities, accelerate research and development process. AI can also complement human expertise and creativity by providing insight, suggestion-based analysis and learning. And this is shockingly accurate. And indeed, hopefully with this lecture, I'd like to show you that AI is not a foe, but it's a friend that can really help us chemists solve the hardest problems that our society is facing. Now, you also have seen, the latest advancements in AI. You see the system like DALL-E or Midjourney that actually can do really cool things with images. And hopefully in this lecture, I can convince you that equally fascinating things can be done with molecules and proteins as well. Now, it's impossible to squeeze in and describe everything in one little lecture. So if you're interested to learn about more technical details about what is the AI and ML methods, and get a little bit open the hood and look for the specific methods. This is about a year ago. It's a review in the Chemical Reviews magazine that shows you different aspects of application, different types of AI and ML methods. So each point here on the right is a specific methods, and you see this review dissects different types of methods and group them and so forth. What I'd like to show you just one little example, that as a chemist, we typically ask different type of questions, right? So is my new theory valid? Is that molecules provide me those properties I'm interested in? How do I make these materials? How do I synthesize the materials, right? Or can I rationalize structure property relations? Now, you can think about and design or use or adapt ML method to answer exactly this query. And therefore, this little table shows you different types of machine learning methods and their logical mapping. So for example, we can use probabilistic methods, things like Bayesian methods, and give us the odds or probability. The theory is okay, is valid. We can use evolutionary methods and find, using genetic algorithms and find the molecules that give us the properties we interest. Or we can use connection-based methods or neural network and, predict how the molecule can be synthesized and so forth. And therefore, there is a method that can fit for the specific purpose. In our lab, we really like the family of methods that called neural networks. So those are methods that are loosely biologically inspired by how brain works. Our idea is similar, right? So your brain have billions and billions of neurons. So here on the left is a biological neuron. So you have the nucleus, you have an axon, and basically when you have a certain chemical process, neuron activates and passes the essentially biological signal through a cascade of chemical reactions, sensing, and transport in the synapses. Now, neural networks in the computations use a simple mathematical model as you have a certain activation functions, you get number of weights, and when the sum of weights above certain thresholds, the neuron activates and passes the output. So this is a very simple parametrical model of the biological neuron. And then what you do, and to have what's called the artificial neural network, you add those functions into the layer. And you see this line such as connections between the neurons. You feed in the input and you get the output. And therefore, this is how you train this model by fitting this different weights. And again, it's a very simple model, nothing that approach complexity of the real brain. So in practice, neural network look like this. This is a piece of the Python code that implements, one of the typical neural network architecture. And there's no magic to it. It's mostly linear algebra and multiplication of matrices and tensors. Now, with that, again, due to the talent and hard work of the student and the postdoctoral fellow in our lab, I think our lab contributed a couple of methods that advanced in AI in chemical sciences. So here on the left is the idea that the neural network can serve you as the hypothesis generation. It can generate molecules with the desired properties, and that work been inspired by the system of the AlphaGo, and I will talk a little bit later in detail about it. And here on the right is another system we're developing that can approximate very expensive solution of the Schrödinger equation. So you solve the quantum mechanical problem, and you can use machine learning to approximate the solution of Schrödinger equation. Therefore, it's allowed you to do the simulation of molecules about 1 million times faster. So instead of running on a large supercomputers, you can run our simulation essentially on a laptop. Now, let me put these things into the context. So for the rest of my lecture, what I'm going to show you different stages of the drug discovery pipeline and highlight some of the methods that other people developed or we contribute. And again, please, if you have any questions, unmute, and I will be really happy to discuss or answer any questions. So this is a standard overview how drug discovery pipeline looks. It's a very long expensive and slow process, can take literally dozens of years. In academic environment at the universities, we typically here on the left side, and we work, in mostly in preclinical. But drug discovery starts from the pre-discovery, finding a disease and a pathway, and then starting, identifying small molecules, usually thousands, doing stages of screening, finding a hit, finding a lead optimization, then doing preclinical studies, optimizing various toxicology, oral bioavailability, other properties. And then, if all things go right, then you go to the clinical trials, when again gets now trying those molecules with humans, and maybe eventually you end up in the FDA approved. As you can see, this funnel, this process is very inefficient. You start from multiple thousands of molecules, maybe end up in one if you're successful. So latest estimates says that for a typical drug to reach your market in your pharmacy store, it may take up to $2 billion. This is a crazy expensive process. Now, how to start? So first step is, the research entities and typically pharmaceutical companies need to identify disease. Again, companies typically commercial enterprise. So the companies look to for disease with a large market. That's why there's very few drugs for a rare disease or a disease that affects small subset of populations. This is where foundations and academic research is typically contributes, but commercial entities typically do, avoid disease with a small market, which is a little bit sad reality. And also, again, for the same reasons, because companies need to be profitable, markets that are lower of the individuals with lower economic status are typically avoided. So disease, which only affect, third world countries, was typically underserved as well. So what are diseases that's typically served? So those are what's called the disease that affect first world countries. So those are cancer, cardiovascular disease, depression, diabetes, flu, migraine, and obesity. Some of this, as you can see, also chronic disease, which means the patient will have to buy pills all over and over again. So those are maintenance drugs, and therefore companies can have nice profits out. Now, when disease has been identified, then we need to think about what to target, and when you find a specific biological target or a pathway. So that can be, a macromolecule, like a protein, or maybe a specific pathway. And those are typically, research, biological studies I run to identify a key pathway in the development of the disease. And therefore, the pathway can be targeted, blocked, or activated. So this disease can be mitigated. Sometimes things get incidental or lucky, from looking at historical data in papers, and maybe some of this pathway or proteins are known before. However, with advancement of the next generation sequencing and technologies like CRISPR, dissecting of the biological pathways is getting much easier. And mapping, because we have a full genome information, full sequencing, with the CRISPR, you can do gene knockouts, and therefore those technologies really drastically accelerate finding a specific proteins to target. However, in the past, a lot of medicine are typically isolated from plants and various animals, and therefore those historical findings were mostly serendipitous. And human eating a leaves of certain plant and finding an effect that was the kind of the earliest type of the incidental discovery of biologically compounds. However, these days, all of the wealth of the information about disease, proteins, patients, genes, and biological activity of molecules are residing in the databases. And we can build a graph, what's called the knowledge graph, and build these associations. So you can see relationship between the graph in the entities. For example, we have a drug, genes, a common disease. So drugs acts on a target and treats a disease through some kind of biological activity. So you can build this relationship on a computer through databases, and through the mining of this association of known associations, or exploring new associations, you can find a hypothesis. What molecule to use, what biological target to treat a specific disease, what gene to affect, and so forth. Additionally, we see the revolution in the structural biology. And you've probably seen the system, what's called the AlphaFold. Now it's a second generation of its structure. So this is a neural network that predicts, a three-dimensional structure of the proteins, given its sequence. That system, for past several years, won a number of competitions to predict three-dimensional structure of the proteins. It now used all over, the industry and academia to predict structure, high-quality structure of the proteins, and therefore, because the protein crystallization is really hard. So very simply, how the system works. So first, what you need to do for your target protein, you need to take your sequence of your protein and perform sequence alignment from with the homologs and orthologs of your protein. So those are proteins, either homologs from the human, or maybe the orthologs from other animals, and or species. And then with the system, with the AlphaFold system, it takes this alignment of the essentially the amino acids that have analogs in the space of the protein homologs and orthologs, and it looks for a database of known proteins, and it creates what's called the contact map. So there's essentially a distance between the protein residues and the backbone of the proteins. So those are pairwise distances between all the amino acids in the protein. And since the distance is three-dimensional, so this essential contact map shows essentially like a fingerprint how the specific protein been folded into three-dimensional space. Now, so those two things serve as the input into our ML system. And it's a very complicated neural network. For the purpose of the lecture, we'll not go into technical details, but it feeds both sequence and these templates of the known crystal structures through the layers of this neural network, and it predicts a trial three-dimensional structure. So what they found out, because biology is so complex, if you do it once, it doesn't work very well. So they have this, kind of a feedback loop. When we have the first prediction, they feed it one more time, and they eventually run it 3 cycles. So like converging the predictions. So here, when you run it first time, system from 0, it gets your first generation dimensional structures. Then you feed it in, you refine it, and doing better and better. And essentially, through this iterative process, the system can predict high quality three-dimensional structures. Now, the way how the system builds, it actually predicts the plane and the relationship in 3D structure of between essentially the backbone of the protein by doing this affine transformations. But instead of boring you with math, let me show you an example of the video. So what this movie will show you is this iteration of prediction. So this first counter will show you the iteration, and inside of each iteration, it has this, when each block runs and predict the structure. Let's see if the movie works. Yeah, you see, you start prediction that's 3D structure. It gets a couple of folds. Again, it's still iteration iteration 1, different blocks. So it refines. Then go to second iteration. You see the refinement of this flexible loops. Structure get better and better. Again, when you feed the iteration 3 at the final pass, you see this is 5 the main protein. This 2 loops here. Again, extremely complex protein get predicted. Now, when the biology has been determined, now it's time to choose the bioassay. So bioassay would be the specific experiment to test our molecules. Again, certain definitions. This experiment can be in vitro, in the artificial environment, essentially in the test tube, can be in vivo, or in the living body, and let's say this your mouse experiments, or maybe ex vivo. Again, those are modern approaches when we have a test on a tissue taken from the organism. And again, as usual, in vitro testing allow us to do throughput. We can run multiple compounds at the speed. However, as you probably know, the in vitro experiment is not always necessary to corroborate experiment in the living animals. Now, on the flip of the coin, if you want to do animal experiments, those are typically more expensive. Also can cause the suffering of the animals, because essentially you make them sick and then you treat them, but you treat only the control group, and the results again can interfere, because again, mouse systems are not as human, so there are also differences between biological systems. Now, as soon as this has been done, now this is where chemistry stinks. So finding the lead. Again, historically, screening of the natural products, plant, microbes, marine world animals provide a really rich source of natural products and biologically active molecules. Finding the penicillin and the salicylic acid from the willow bark tree. Those examples of natural products been translated as the drugs. Now, a more modern approach is to use someone else's lead. And there are a number of companies that takes existing molecules that have been approved and try to do clever chemistry, essentially avoid patent litigations and change a molecule just enough that make it active, but also not similar enough so not to get in trouble. Sometimes, again, this practice may be ethical, maybe not ethical, but also can lead to dramatic improvement in biological activity. And what we see, different generation of, for example, antibiotics have much better activity than original discovery. So again, you get pluses and minuses here. And traditionally, how the screening is done in the pharmaceutical companies, they have a collection of thousands of molecules, typically stored in these vials and freezer, and those molecules are screened depending on the new target, and this is your starting molecules. And this is where essentially computational chemistry kicks in, because now we know the structure of our receptor, and we have a collection of the molecules, and we can use computer to essentially drive this process, and therefore maybe we can design a molecule in the computer. I'm pause here for 1 second, and is there any questions so far?
Have a technical moment, Alexander. There is your sound is quite noisy, so if you can fix this, that would be good. But if not, we can hear you. But
okay.
How about this?
Oh, much better, much better. Thank you.
Okay. Let me share back my screen. So if there are, oh, sorry. Can you see my screen now?
Yeah.
So if there are no questions, I actually have a quick question to the audience. And so a chemical company that could synthesize most organic molecules in the world located China, Germany, Ukraine, or United States. Does anyone know an answer? Any brave students?
I know the answer, but perhaps
Yes, yes.
Right.
Some of the older people know the answer, but I'm asking the young people.
Okay.
No brave person? And this is the example when we can actually be proud, because it is located in Kiev. It is Inamin. It's, I think, one of the scientific success stories and fantastic companies who many pharmaceutical companies in the world are grateful because of the technology of synthesizing ultra large chemical libraries. And this is a screenshot from last year when the size of the Inamin libraries were 30 billions of molecules. And this year is already 40. So it's double less than a year. Now, since imagine those are billions of molecules, essentially the problem what the chemists are facing is almost like navigating the galaxy. But now, instead of the stars in the galaxy, each star is a molecule, and now chemists need to navigate and find that particular star that, for example, has an Earth and has a life. Therefore, this is a very complicated problem. Now, how AI can help us to mitigate that problem? So traditional drug discovery projects use what's called the virtual screening. And the idea is very simple. When you start with your library or database of molecules, typically, millions, now it's billions, and they use, a computational method of virtual high throughput screening to identify a small number of molecules, what we call hit, to prioritize for experiments. So you sift in very fast, large piles of molecules to find few those hits. And this is the problem of finding a needle in the haystack. Now, the problem is it's very inefficient. And as the library is getting bigger and bigger, this haystack is larger and larger. Therefore, it is hard to find those needles. Now, what AI can offer us is this intriguing idea. Can we generate needles directly without going through the haystack? And the answer is yes. And the technology, what you probably have seen, is what's called the deep fakes. But the specific under the hood is the type of neural networks called generative adversarial neural networks. And idea is that you can take any photo or an image of a person, and you can copy a style. For example, you can copy, a hair, a glasses, a smile, skin color, and so forth. So you can take style of one picture and copy it to another. You probably have seen a lot of apps on your cell phone to make your pictures in a style of your favorite painter. So this is like a Van Gogh, a picture of the car, and so forth. So those types of neural networks that can generate new type of image and depending on the conditions. Now, mathematically, again, very simple. Those methods, predictive versus generative methods. So predictive methods in language of chemistry would be virtual screening. Generative methods in terms of language, what's called the inverse design problem. Mathematically, they describe different probability distribution functions. P, labels given our molecules. So Y would be our numeric property of biological activity, solubility, so forth. So the predictive methods, given a molecule, predict a label. So what's happened, you take molecule, pass it through your machine learning systems, and you predict properties. Now, the advantage of generative models, it models probability of molecules given their properties. So you tell a system what is your desired properties, and it comes up to you with the hypothesis of molecules. So you flip the whole pipeline. In terms of that, chemists designed multiple types of those models. Again, so this is 2D depiction of a typical organic molecules. You can use different types of representation. You can use one-dimensional representation like a language. So you can encode molecules into the language of smile strings. So it's essentially characters that encode two-dimensional structure. You can work directly in molecular graphs and ask a system to build molecule by placing atoms and drawing the bonds, or you can also be very sophisticated and make it three-dimensional structure, because molecules are three-dimensional objects. Now, again, I'll show you brief examples with the use of smiles. Again, smiles is essentially a language of encoding 2D structure molecules in the string of characters. So it has a syntaxis and its own grammar. So what we ask a molecule to learn this language, but instead of the, as a normal language, as Ukrainian or English, it's a language of encoding chemical structures. So if you're interested to learn more, this is an excellent review in Science by some of our colleagues in University of Toronto, and it goes through different types of these molecules. But let me show you one example of the system we developed about 5 years ago. Excuse me. The system takes inspiration from this a system that essentially play the game of Go, the AlphaGo system. The system moves checkers on the board, and it uses what's called the reinforcement learning. So essentially, it's AI agent that move checkers on the board and essentially play the game, so interacts with the environment, with us, physical world. And as a reward, the system receives essentially if it win or lose the game. And the idea is that game of this agent learn how to maximize the reward. Therefore, it's essentially master to play this game. And by this time, those systems are almost ubiquitous. You know, they play video games, StarCraft, Warcraft, they play competitively, and they can drive cars, and you know, they can drive robots and so forth. But for purpose of our game, you know, our neural network draw a molecule, then we estimate the properties of its molecule, and that property can be, you know, affinity of the molecule to a protein or a physical property or combination, and we feed it back to inform a system. Is it molecule good or bad? So this is how it works. Again, we ask neural network to generate sequence of smiles. So the sequence of smiles corresponds to a molecule. Now we have another system, which is predictive model. So it's like an oracle. It's a model that predicts properties of that molecule given its structure. So we feed these molecules into our predictive model, and the model says this molecule is inactive, so it's not very interesting. So then what we use, our reinforcement learning, our stick, and we say, hey, neural network, that molecule is not good, so do better. And you know, in reality, there's a mathematical algorithm that we can change the weights of the neural network into direction to change this molecule be more active. And you iterate, many, many times until the next time when the molecule generated passed through a predictive model, and now it became active. So then we use a positive reward. So here is our carrot. And eventually we have a happy model. So let me show you a little movie. And the beginning of this exercise, so what this plot shows you, this is the probability of the molecule being active. And you see in the beginning, everything at 0. So everything is inactive. But as you start to train the system, you see this probability shift, and more and larger fraction of molecules get active. And here is a probability of the molecule binding to a specific receptor. Now, as a chemist, you can play different games. You can make molecules more complex. You can ask system include a specific substructure. So for example, here is the game. We ask a system to add more and more benzene rings. So it became, you know, more and more complicated, but still realistically looking molecules. And now let me show you a real example from the paper and collaboration with the Structural Genomic Consortium in Inamin. And our medicinal chemists were interested to prediction and finding a molecule to inhibit a specific protein kinase. So kinases, those are signaling proteins. You probably know they implicated cancer, inflammation, Parkinson's, and you know, it's one of the largest and widely studied family of the proteins. So here, and this is the reference for a specific paper that published last year, we ask a system, and we play this again, little game. We ask a system to compete with the computational chemist to develop essentially an inhibitor of the protein. And again, if that lecture would be in person, I would poll you, you know, maybe if someone wanna predict who would win, if the human chemist or robot would win the competition to design inhibitor for EGFR kinase. Anyone wanna make a bet? No? Okay.
Maybe human.
Huh?
Maybe human. Still.
Okay, it's a good guess. Let us see. So here is what we ask. We ask a system. We had a budget of certain amount of money. We ask a system selected a number of hit molecules, both positive and negatives. Molecules were synthesized at Inamin. And then we tested them in first enzymatic assay, doing single dose experiments. And for molecules being active, we follow up with essentially 10 dose and do full IC50. And for certain, we also did some more cell-based assay. So this is the pipeline. We get a budget to buy 30 molecules. We ask system do 25 for 5, 25 positive hits, 5 negative hits. Again, only 25. We have to be grateful how great enamine is. 25 were synthesized. Unfortunately, 3 were insoluble, and solubility control was not part of this experiments. So 22 molecules were tested in EGFR. 5 inactives were all confirmed as inactive. So this is all good. 4 compounds confirmed and measured. So we have 27% hit rate for the autonomous system. I would say this is on par. So we have a tie here, on par with essentially, traditional human, by human guided molecule selection that is essentially guided by the experimental medicinal chemist. But the study shows that you can properly train this AI system and learn useful medicinal chemistry. So that system can help us to select better molecules and also be better reproducible. Now, this is another study that we use different approach. And again, for past 3 years, we all stuck with the COVID. And this is another study that ML helped us to generate and produce the molecule to inhibit one of the protease. It's papain-like protease of the SARS-CoV-2 virus. Again, study was published recently. But interestingly, not only AI system were helping us to design molecules. So here on the left, this paper is also on the cover of GCM, so Journal of Chemical Information and Modeling. And that cover was designed by one of this AI stable diffusion system that can generate images. And then essentially this is impression of the robot pointing on the molecule in the vastness of the chemical space. Now, in the last few minutes, let me switch gear a little bit and go back to idea of the virtual screening as kind of more traditional directions of define. So here where in many studies, you maybe want to consider explicit protein ligand interactions. When you take a 3-dimensional structure of the protein ligand, you dock it and you understand interactions between ligand and a molecule, you find compute binding affinities and so forth. Now, unfortunately, when you do docking, and people who do medicinal chemistry project knows that docking has a lot of false positives, not very accurate, but it can get you interesting hypotheses. Now, if you really want to understand how fundamentally molecule binds, because protein molecules, quantum objects, you need to solve Schrödinger equations. So because all molecules are quantum objects, this is your essentially quantum mechanics 101. It's time-independent Schrödinger equation. But from the point of view of us as the data scientist, you can formulate it as the machine learning problem when the energy of the system is this function f depending on the molecular coordinates. And again, so what we do, we take molecular structure, feed them to inside the neural network, predict the energy and other various properties. So why machine learning is really helpful? Because of that problem. So we can do this very simple exercise that if you're interested to study large system, you have to use approximate and fast method, things like docking and the force fields and molecular dynamics. But those are very approximate crude methods. And if you really want to improve the accuracy, you typically need to use more quantum mechanicals, more expensive approximations. And here on the x-axis, we have the scale of the method. You start from a linear scaling methods like a force fields to order 7 methods, which is couple cluster theory. Essentially, you can compute on the small molecules. But the beauty of and the promise of ML is that you can bridge this gap and you can use these expensive calculations to train this ML model and run the simulation much faster. And again, the contribution from our lab a few years ago is what's called the ANI, Deep Neural Network framework. Those are systems that compute, energy and properties of a small organic molecules. It's about 1 million times faster than traditional quantum mechanical calculations. And these types of methods these days are quite popular and used in many projects. So again, if you go back to medicinal chemistry example, one of the way how to compute protein ligand interactions into do what's called the free energy simulations, when you compute explicit simulation of the ligand in the pocket of the active site of the enzyme, and you do molecular dynamic simulations, and you understand and compute free energy of that simulations. Now, the simulation are done with the empirical methods, what's called the force fields. Now, in this paper, what we did, you can replace just one part of your system, a ligand. That's what we've done so far. With our neural network, they can essentially predict the structure, energy, and properties of this small molecule, and the rest is still done as usual. So we replaced just a little part of the system. Now, what it shows us that here on the left is the comparison of an accuracy of the predicted free energy of protein ligand binding for the specific enzyme for the, for this congeneric series of molecules. You see they have the same scaffold and different R groups. So this is the typical accuracy of these simulations. However, what you want to see if you run realistic projects, this accuracy may not be enough because there's a number of molecules that are outliers. This is because of the approximations. So if you replace the part, just the ligand part, with the neural network, we see here on the right, immediately, errors are twice as smaller. All your molecules are inside the shaded area, and the shaded area, this is, the desired of accuracy, 1 kcal per mole. And indeed, we see reduction of errors and drastic improvement of accuracy using this ML methods. So with that, let me finish, and I think just tell you kind of my vision where all of this goes. That with advancement of AI and automated, I think where chemistry is going, is that we will have this AI-based system when you can ask AI for useful hypotheses. For example, a system like ChatGPT, you can write it, or with your Siri assistant on your iPhone with a voice. So this AI system would be able to get your hypothesis and maybe run a computational simulation to make this hypothesis better. And then eventually we want to connect the system with the high throughput synthesis and high throughput experiment and robotic characterization, is a physical properties or biological activity. So in my opinion, one of the direction of chemical sciences is going to automated science and autonomous discovery. Essentially the Edison, when he was looking for a filament for the bulb, was exploring a lot of different materials, and that was essentially a serendipitous discovery. And now we can truly run this discovery projects with the this help of the automation. And with that, there is a new initiative at the Carnegie Mellon. So we're building this fully automated facility with 200 different instruments. starting from synthesis, purification, characterization of material, HPLC, NMR, you name it. An idea is that everything is going to be controlled with the code, and you can run experiments and hopefully democratize access. As the remote facility, you don't need to go to the lab. You can run your experiment at the comfort of your home. And hopefully this will help us to drive next generation of drugs and materials. And with that, many thank you for attention. Дякую, пане Олександр, за and
I see that there are some questions in our chat already.
ми можемо українською.
Добре, добре, так.
Okay, so the question says, what software do you usually use specifically for screening, docking, creating needle, chemical design? Maybe it's sorry. We write most of our software in Python. So we use a couple of Python libraries to essentially run these workflows. All of our neural networks are developed on our own, and all the code available on GitHub, free. If anyone interested, I'm more than happy to show, connect with my students in the lab and show how to use.
Thank you. Are there any other questions?
Yes, I have one question. Thank you very much for your presentation. And my question is, so you need like to write a new code for each new task, or you have some general programs? How it,
it's a fantastic question. No, you don't need to write a program. What you need to write is a piece of the task. So we provide pre-trained model that generates molecules. What you need is just to tell the system you knew protein and train a predictive model for this protein, because we don't know in advance which protein, for which project is needed. So you need to essentially write just a little simple function for, new task.
But this function should be new for each protein, right?
Yes, because you need to tell a name of the protein, a structure, and historical data.
Okay, thank you.
Thank you. Are there any questions? No more questions? I just see some small discussion with students on the fact that it is Ukrainian company that produced most compounds in the world. I was surprised that they don't know this fact. In fact, but it seems,
what a shame! What a shame!
I have to correct that. This in my lectures, I think. I have one more question. If there are no other questions. Not about, less about medicinal chemistry, but more about general organic chemistry, for example. What is the state of the art with predictions of efficiency of organic chemistry reactions at the moment? Say about that.
Yes, I owe you a paper. I know. So that's the short answer. The long answer. Yield and outcome is hard, and my take on that is these gigantic databases from like Reaccess, speaking blindly, there are not very high quality of the data, and therefore it's not very useful to use essentially data mined from literature to predict yields. I believe the solution to that would be twofold. The new data that is generated in the controlled conditions, things like either running a robot or like Inamin doing essentially one type of experiment, and it's a controlled environment. And the second, what we are trying to do is this with fast quantum mechanical methods. We can, knowing the mechanism, we can predict transition states and we can predict the outcomes through a mechanistic hypothesis.
Thank you. Are there any other questions? So that we have no other questions. So I'd like to thank Alexander for his very nice talk today. And I hope that it was inspiring for our listeners and for those who will be seeing it on YouTube. I will place this on my YouTube channel if Alexander doesn't mind this. And with that, I would like to.
Також, коли ми виграємо цю війну, я також сподіваюся, що студенти захочуть поступати до PhD до нас або до інших університетів, навчатися цим технологіям та методам. Так що, якщо хтось захоче поступати до нас на PhD на хімії, computational biology, computer science or structural biology, то я буду радий допомогти. Тому що нас, на жаль, дуже мало українських студентів.
Дякую. Дякую ще раз Олександру за його чудову лекцію і до зустрічі. До зустрічі наступного разу.
Дякую. До побачення.
До побачення, гарного дня.
Дякую, до побачення.